Angelica Muñoz. I worked here at the Chicago Botanic Garden as an intern this past summer. I worked here at the Plant Science Center for the past 10 weeks doing some awesome research with great people. Let me tell you what I did. My research involved two species of flowers from the Onagraceae family, commonly known as the evening-primrose family, which are Oenothera gayleana and Oenothera hartwegii, subspecies filifolia. What is interesting about these desert flowers from the American Southwest is that they are said to be incompatible with themselves, which means they shouldn't be able to reproduce with its own pollen on the same plant, otherwise known as selfing. These flowers may look similar, but they have key differences that make for interesting additions to our experiment. Some of the differences is the size of the inflorescence, where filifolia is larger than gayleana, the tube length, where filifolia has a longer tube than gayleana, leaf form, where filifolia is thin and long versus bushy, and pollinator syndrome. Gayleana is pollinated by bees and is unscented, while filifolia is pollinated by hawk moths and is scented. This then led us to wonder what effect different treatments of selfing pollination, either autogamous pollination by the pollen of the same flower onto itself, or gytonogamous pollination by a different flower on the same plant. We also have outcrossing, such as pollination with a sibling from the same mother plant, or within pollination with a member of the same population and between populations, where pollination with a member outside of a group is also done. To begin the experiment, the crosses had to be done to each plant. Then we waited for them to grow with the help of fertilizer, water, and sunshine. Once there were flowers, which only last open a day or so, we used a caliber to measure the length of the petals, the stamen, the anthers, the nectar tube, and the floral flare. In addition to all the morphology, we set up pollination stations to observe if in the case of Oenothera gayleana there was a difference in bee visitation or bee type for the various crosses. To do this, we would monitor and record any visits to individual flowers in 40-minute increments. After running statistical analysis, we see that fitness does vary depending on our cross types that we